This is Boston University's Neurophotonics podcast. My name is Sung An, a second-year PhD candidate in Biomedical Engineering, and I am the producer and editor of this episode of the podcast. This episode, our enlightened hosts are joined by Dr. Irving Bigio as he explains his long and amazing journey through the field of physics and exciting adventures in biomedical imaging. They talk about polarized light, birefringence, and how to stay motivated in science. For our listeners, I would like to personally apologize for the less than ideal sound quality due to difficulty with the mic settings. Additionally, as the topic today is inherently visual heavy, please watch the video version if you are able to enjoy all the pretty pictures. In any case, thank you for tuning in, and please enjoy the episode.
Hello, everyone. Welcome to another episode of the Neurophotonics podcast, created by trainees at the Neurophotonics Center here at Boston University. My name is Dora Balog. I'm a third-year PhD candidate in Biomedical Engineering, and I'm here today with my co-host.
Sulam Duwali. I'm also third-year BME PhD candidate and Neurophotonics trainee as well.
Today, we are here with Professor Irving Bigio, who is a professor at Boston University with appointments in biomedical engineering, electrical and computer engineering, physics, and the BU School of Medicine. He has authored over 200 scientific publications on laser physics, nonlinear optics, and biomedical optics, including the award-winning textbook Quantitative Biomedical Optics. Dr. Bigio has pioneered the development of a number of biomedical optics technologies and tested them in preclinical and clinical studies. Clinical diagnostic methods based on his development of elastic scattering spectroscopy have been developed commercially for clinical practice and have received FDA clearance. More recently, his life has been investing in quantitative birefringence microscopy to image the underlying pathologies of the brain that are associated with a variety of neurodegenerative diseases such as Alzheimer's disease and age-related dementia. Welcome, Professor.
Thank you. So I guess people like to start with knowing where I came from and how I got here. And since I'm old, that's a long story. I started off becoming enchanted with the physics of light when I was a teenager. I was growing up in South America, in Colombia, and I went to the American Embassy in their library, and I'd like to look at the Scientific American issues there. And there was this issue of Scientific American that was announcing the invention of the laser, the first demonstration of the laser. This is back in 1860 or 61, 60. And I was totally fascinated. I became totally in love with the physics of how things work that make light, detect light, use it to image, use it to measure. And so when I then went to the States to go to college, I studied physics and focused on optical physics. So I did all of my degrees at the University of Michigan, which was actually the birthplace of nonlinear optics. The first paper ever published in the topic or field of nonlinear optics was published by professors and a graduate student at the University of Michigan. One of those professors was my co-thesis advisor. So again, at that time, it was pretty much pure physics and the physics of lasers and light and nonlinear interactions of light with matter. And that was the basis of my PhD thesis. And so it was not unexpected or unusual that my first job was using my expertise in laser physics and optics to apply to physicsy kinds of things. So my first job, I was fortunate. I got a well-paying job right out of my PhD at Los Alamos National Laboratory. If any of you have seen the movie Oppenheimer, you now know what Los Alamos was, at least back then. After the war, it became a more general national laboratory, not just nuclear weapons. And when I joined the laboratory in 1974, fresh out of school, they were building two large programs. One was laser fusion, for the ultimate idealistic source of energy, fusion energy, and the other program was laser isotope separation. Can we come up with cleaner ways of separating, let's say, uranium isotopes if you wanted it for nuclear power, or other isotopes for medicine or other purposes? Rather than having to use nuclear reactors to produce them, can we do it with light? So those two programs appealed to me, and at the time I sort of had the right background at the right time in the right place, and so they were happy to hire me, and I was happy to move to Los Alamos, which is a very lovely place to live and raise your kids. It's high altitude in the mountains at 7,300 feet altitude. There was a ski area 15 minutes from our house. The house we eventually built on the side of a canyon. We had designated wilderness behind our house, so nobody could build there, but we could hike there. And of course, raising our kids in the mountains was a wonderful place to be, and a place where you didn't have to worry about crime. And the school was filled with the kids, PhDs, and so that your kids had a great school to go to and cliques of other students to interact with. Anyway, I stayed there for 26 years. It was my first job and the longest job so far. And then after our kids were off to college or gone, at that time, I decided to take myself out of management. I had been managing the Laser Science and Applications program there. And was the director of the Laser Science and Applications group. And we were doing all kinds of laser optics, optical physics things, very physicsy things. But I became fascinated in the applications of light then in biological questions. It started off at a very basic science level, trying to understand nonlinear interactions between light and biological molecules. And back then, there was a very controversial thing going on out there that someone had published a paper in Physrev Letters that said they had detected nonlinear waves in DNA molecules. And I looked at it, and I studied the paper, and I said, well, it makes lots of sense if you use just the theory that we know from physics, but they seem to have forgotten something. And that is that these biological molecules in nature are surrounded by water. And when a biological molecule like DNA or protein is surrounded by water, these nonlinear excitations in the molecule are damped out because the water is always attached to the surface and several layers of water. And so I went about working with theoreticians there, and we did experiments in my lab to prove that the publications that had come out were experimental artifacts to be claimed about it.
So that experiment was still at Los Alamos.
We did experiments at Los Alamos. The original research that was published was a combination of stuff that came out of Russia and stuff that came out of the University of Maryland. And so this was a big controversy at the time.
Yes.
It was interesting. At first, there was an editorial in the journal Nature that said, "Have the physicists hijacked our molecules?"
It's sketchy.
Written by a very famous bioscientist, Richard Maddox. And then when our paper came out debunking the original publications, there was another editorial in Nature that came out, and it said, "The physicists retreat with their tail between their legs," or something like that. It was, the curious thing about that was that editorial was written by a physicist who was here at BU, a professor in Biomedical Engineering department at the time, named Maxim Frank Kamenetsky. And so I came to BU. So what happened was I got more and more involved in biological applications of light and lasers and things like that. And I decided I was really more interested not in molecular biology, but in things that could actually be useful in clinical applications to actually have an impact on the human condition and translational things. So I started, and what I was good at was things like spectroscopy, things like that, and understanding how light interacts with matter. So I started working on topic, which eventually we called elastic scattering spectroscopy, as a diagnostic by studying the scattering properties of tissue, which relates to the size and density of scattering centers, the nuclei enlarged or is the chromatin condensed or what's going on in the tissue, and found that we could do a good job of correlating spectral changes with disease state. And we started doing some work with collaborators at other institutions elsewhere because we didn't have a clinical research center in Los Alamos. And then I realized that I really need to be closer to clinical research centers where I can collaborate directly with the medical research scientists. And just at that time, two things happened that provided impetus for me to leave Los Alamos. One is that I was interviewing at various universities that were interested in biomedical optics and building programs of biomedical optics in their B&E departments. I was interviewing them. And the second thing that happened was that there was a forest fire that came through and hit Los Alamos. And my house was one of the 400 houses that were burned. And so we were living in temporary housing after that. So it was another impetus to leave. And serendipity, an old friend of mine who had been the chair of physics at the University of Illinois, was coming to be the new dean of engineering at Boston University, David Campbell, who eventually became provost at Boston University, but now is a physics professor in university. He called me up one day and said, "Irving, I've accepted a job at BU. I'm going to be the new dean of engineering there, and I'm going to recruit you there." I said, "Oh, I didn't. BU wasn't on my radar, but I came out to visit, and I met various faculty, both in the DME department and in the medical school, and I was blown away with not only how brilliant they were, but how collegial they were." And I said, "Okay, I could be happy here. And if I want to do clinical research, Boston is kind of Mecca, right?" Because not only our own medical school, which was really trying hard in those days to build research portfolios and interact with the Charles River campus, but also, of course, the various Harvard hospitals in the area, where lots of people were kind of becoming interested in biomedical optics. So that brought me here to BU and got me more directly involved in clinical translational research and developing technologies that are useful in the clinic. And so the rest is history. As Dora was mentioning, one of the technologies we developed, elastic scattering spectroscopy, which basically is so simple you can't believe it works. You have two optical fibers, you shine light on tissue, you look at the spectrum that comes back. You shine white light, you look at the spectrum that comes back, which is not so white. And from that, you extract information about the diagnostic pathological state of the tissue. Now, you can use all kinds of models, and in my class, I teach those models of how scattering changes with particle size and density and things like that. But it turns out that you're going to be able to model certain features, but not everything, because you don't know everything that's going on when light scatters back from tissue. You don't know what's the mix of chromophores. You don't know what's the mix of the size distribution and whatnot. So you can do your best to model it, but actually, it was a very nice opportunity to apply deep learning and AI methods. And so, still at Los Alamos back in 1999 or 2000, we published our first paper on using AI methods. We compared 2 or 3 different methods for extracting or correlating diagnostics with the scattering spectra, correlating that with pathology. That ended up working. And it took 20 years longer or more for this kind of thing to make its way through FDA. But the company called Neuroscience licensed my patents here at BU and was the first FDA cleared technology using AI for the primary care setting AI has been used, for example, to assist radiologists with mammography, but it's never been approved as being the main element of a clinical device that's intended for primary care. So we're trying to democratize that, bring the cost down and the usability, make it very user friendly, so a nurse practitioner can use it, or even a nurse doesn't have to go to a specialist. But to know when to use its primary application for dermosense, so as the name might imply, is for skin cancer screening. And so patients can go to their primary care physician and take off their shirt, and they've got a bunch of things on their back, and the doctor can check them out and see which one might be risky. And if the device tells you it's high risk, then you send them to their dermatologist. If it doesn't say high risk, then you say, see you next year for your next physical. So that's up and running, and there are other clinical applications that we continue to collaborate with physicians and surgeons at the medical center, using scanning spectroscopy as the diagnostic. But along the way, I also realized that there are other aspects, more physicsy aspects of light interacting with material, with medium, that we hadn't explored adequately, and that was using the polarization properties of light. And I had always been interested in neuroscience, even though I knew nothing about neuroscience. But I knew that there are some things I could image in neurological tissues that are difficult to otherwise image using polarized light. And that led to my current passion, which is in using polarized light to image brain tissue, in particular to image myelin and its structural integrity or lack thereof, and relate that to neurological diseases. But also just to understand the brain, so helping people who are very interested in connectomics, where the axons from this side of the brain go to that side of the brain and how they get there, to understand basically how the brain works. Again, I'm not a neuroscientist, but we have some tools that are pretty powerful, and so we collaborate with exciting projects with collaborators in the medical center and elsewhere on neuroscience problems. And you get your name on really cool papers, even though you don't know any of the neuroscientists.
So that's an interesting transition into neuroscience, right? Like you have background in electrical engineering, laser physics, and everything, and at BU you got the opportunity to more neuroscience. But would you mind expanding more on how you landed that opportunity, the first collaboration at BU for neuroscience purpose? If you could expand more.
Sure. Well, it started off with just doing some things on our own. And I could probably show some of that with slides. So what you're seeing here, by the way, is an image of human brain tissue from the brain bank at the medical center. These are patients who either normal controls or patients who have chronic traumatic encephalopathy. BU has the brain bank of NFL football players. And what you see here, what's colored, the color indicates the orientation of the optic axis of the myelin, which is 90 degrees to the direction of the axon. And so these are axon tracks, and you can see how different tracks are crossing each other. The green ones are sort of going up and down, and the purple ones and red ones kind of going horizontal. And you can see when they change direction and they branch and they go into different directions.
Yeah.
And so if you cut a lot of slices and you track all of this, it's a beautiful way of extracting the connectome, which we're hoping to do. That we're trying to figure out how we can store 10 to 20 petabytes of data, which is what it will require. And so we're working on finding someone in Silicon Valley who's willing to host something like on the order of 20 petabytes of data storage for us.
That's a lot of data.
But at the moment, what we're more focused on is neurodegenerative diseases and treatments for neurodegenerative diseases. So I gave a grand rounds talk at the medical center, and one of the shining neuroscientists at the medical center, Professor Douglas Rosine, saw my talk, and we connected and built a collaboration. So that's one of the collaborations we have. We've had a couple of grants funded and doing some really exciting work, studying the effects of stroke and treatments of stroke. And then at the same time, I built a collaboration with Professor David Boas here at the Neurophotonics Center, who's mostly famous for things like FNIRS, but has indeed been interested in neurodegenerative diseases also, imaged using polarization sensitive OCT, can polarized light, and what one of the magical things it can do. And so I started building a collaboration with David and the various neuroscientists that were associated with things that were being happening at the Neurophotonics Center. So Professor Ann McKee, Mike Alonso, people like that at the medical center, who are now becoming our collaborators in grants that we're submitting. We had a Kilochan Fund grant funded to get us started to get some of the preliminary data. And so now we're submitting NIH grants based on the data we extracted from that Kilochan Catalyst Award. Who knows what's happening with NIH, so.
Yeah, big question. So I guess my question related to birefringence and what we're looking at here. So we are looking at brain slices, correct? So how could you tell us a little bit more about the big picture of how brain slices can connect to clinical applications and like how we can learn from them and translate it back into living patients?
Absolutely. So that takes going back to the beginning of these slides. So this is what my current research is about. Imaging the structural details of myelin, and it turns out more than myelin, also cerebral arteries reveal all kinds of things about their nature with polarized light microscopy.
Quickly for our viewers who do not know what polarized light means, would you mind?
We're going to get
on to that. 70 days.
Okay. You win the quarter for the best question. Okay. So first, let's talk about biological tissues. Most biological structures are anisotropic, meaning they're different in one direction than another. Right? That gives them structure. That gives them function. So things like collagen, we know comes in fibers, so it looks different in this direction than transverse. Muscle fibers, lipid bilayer, which by the way is what myelin is made of. It's also the boundary between cells and everything else. And because of that, the optical index of refraction is different for different directions, of the electric field. So the way light interacts with tissue is through its electric field. And electric fields, as a physicist who studied optical physics, is something that I know fairly well. So you go after your strengths. And so I have a pretty good idea how light will interact with birefringent media. So this is just physicist drawing showing you that the index of refraction in one direction might be different than in another direction. And so what this means is, for example, if light is propagating, let's say along this axis, which is the optic axis of a medium, its electric field, which is transverse to the propagation direction, will see a uniform refractive index. If it's propagating in this direction, however, the electric field in this direction will have a different index of refraction than the electric field in the transverse direction. And you can have positive birefringence or negative birefringence. And this is just the underlying physics of why the birefringence relates to the anisotropy. It's because the mobility of electrons in these molecules is different along the chain than transverse to the chain. You might imagine that in these anisotropic structures in tissue, the mobility of electrons in the electron cloud is different along the length, let's say, than transversely. And however that electron cloud is mobile, the more mobile it is, the higher the index of refraction and the slower light propagates with its electric field at that axis. So these pi electrons that roam the chains are the source of this birefringence. Okay, so something about polarized light. So unpolarized light has random directions of its electric field. But linearly polarized light has its electric field in one direction. It doesn't have to be up or down, or it need to be any angle. But it's only in one directional electric field. Circular polarization is what happens when the X and Y polarizations are out of phase by 90 degrees.
Just a quick question, or like relation to maybe polarized sunglasses. So that is filtering out light in the same way, correct? Where you, for example, would be able to see through some surfaces that are very reflective if your sunglasses are filtering out the light.
Right. So if you have polarized sunglasses, you might be interested to know that the blue sky the light is polarized. So how do you convince yourself of this? Put on a pair of polarized glasses. Let the sun be at right angles to you. So don't look at the sun. Look to the side from the sun. Look at the blue sky that the sun living, and then turn your head sideways like that, and the sky will get dark. The blue sky will get darker blue because the light that's scattered from the upper atmosphere is partially polarized. And in order to understand that, you'll have to take my course. I just want to show one thing. Okay, so this shows linear polarization and circular polarization. And so what happens in circular polarization? So here's linear polarization at 45 degrees to your horizontal and vertical planes. It's still linear because the X field and the Y field are in phase. When they're out of phase by 90 degrees, then the electric field just goes around in a circle. That's why we call it circular polarization. Okay, and this is just a little bit more illustrations of how you make circularly polarized light. You purposely delay one axis compared to the other axis. So they become 90 degrees out of phase, and then you end up with this circularly polarized light. And it turns out that things in the nervous system are birefringent, and so they will cause the polarization to change. So here's parallel polarizers. I just took two polarizers and I made them parallel, light goes through. When I cross them, it goes dark. There's a little bit of reflection of the camera that was taking the picture there. But if I put a nerve fiber between them, it lights up. So that was the very first experiment we ever did, and that was with a lobster walking leg nerve. And you can do that at home too. You get two pairs of polarized sunglasses, cross them, one like this, and take a piece of plastic bag and just kind of stretch it and put it in between. You see all kinds of pretty colors. Because when you stretch this polyethylene, you align the polymers and it becomes birefringent wherever you stretch it. And the different colors happen to be the wavelength that has the right amount of rotation to get through the two polarizers. So the first thing we did was we wanted to actually make a movie of an action potential propagating in a nerve. And so we imaged the sciatic nerve from a mouse. You stimulated one end, you put it in a chamber that has electrical stimulation, but allows it to the fiber to sit through between it and windows so you can look at it optically. And just to show you what the structure looks like, we put the nerve in between with a window there, and we stimulate it, and then we can tell when the ether signal arrives at the other end. So we know we stimulated an action potential because it arrived at the other end, which we pick up electrically. But in between, we get an image of the light that goes through. And you can see here's the ethers. The red thing here is the actual electrical signal. It's much shorter than the optical signal. This optical signal has a delay and has a long recovery. Okay. Then we realized that what we were doing is we were looking at a nerve fiber bundle that had axons of all different diameters, and different diameter axons propagate at different velocities. So we then went to a subset of axons, and we got a much faster optical signal. And basically following the ethos signal quite nicely. And then we decided we want to make a movie about how it propagates. So cameras, a typical camera will not go at thousands of frames per second, and at least not unless you have $1 million. But we could get a linear array, a linear photodiode array that could be framed at 5,000 or so frames per second, so we could capture the propagation. Because since the nerve is like a line, we could just focus this line onto the detector between cross polarizers. And when we stimulate it, this is what happens. You actually see the action potential fly through, and then the recovery time.
That's fascinating.
Okay, this is fun. What we really wanted to study was birefringence of neuronal structures to see if we could understand neurological diseases like Alzheimer's. And we want to image them microscopically, not this whole nerve. Okay. So myelin is a lipid bilayer. It's wrapped many times around axons, and it has exquisitely birefringence to it. The optic axis is like that. And it's due to oligodendrocytes in the central nervous system, or Schwann cells in the peripheral nervous system, wrapping the axons with myelin. And this is what you would see if you looked at a cross-section of a myelinated axon with an electron microscope. And you can see that this one is partially damaged because there's a bit of delamination going on there. And that's one of the things we're interested in. But electron microscopy can only look at a very tiny amount at a time. And we wanted to look at whole brain sections, the entire cross-section, let's say, of a monkey brain. So, again, a little bit of explanation about oligodendrocytes and what they're doing. There's a healthy-looking axon mostly. So here's what's going on. So this is the myelin sheath. This is physicist cartoon of myelin around an axon. And because the optic axis is always in that direction, if you have light coming from, let's say, from the top coming down through it, it's going to be crossing the optic axis here, but parallel to the optic axis here. So it'll be sensitive to the edges, and the light here going through the middle won't be changed. And so we'll be able to really image the edges very nicely if the light is coming in that direction. So again, cartoons of what happens with damaged myelin. And here you see this is a bit healthier and when it's lower in the right. And so we built a microscope to do this in a highly controlled fashion. We wanted not just linear polarizers, we wanted to really control the elasticity of the illumination light and the detection light, because we wanted to get out more features of the information, not just through an image of quantitative measures of things. So we built a microscope wherein we could control the relative phase and the polarization of things. This is how you make a circular polarizer. You have a quarter wave plate and a linear polarizer that are 45 degrees to each other. Or we can just simply remove the quarter wave plate and rotate linear polarization and image to the circular polarizer. So this will cross-circular polarizers will produce exquisite qualitative images, and it'll do it wide field instantaneously. So that's another thing about birefringence microscopy, is that it's wide field. It's not a scaling point like you would have in confocal. So you can image large areas of tissue and then tile them together and get whole brain sections rather quickly. So this is just shows what happens if you adjust the focus knob going through, let's say, a 30 micron thick section of in this case, it's gray matter near the white matter boundary. And so different axons come into focus at different planes. This is cross-circular polarization. So each pair of lines is an axon, because we're seeing the edges.
How does your research approach change depending if you're looking at gray or the white matter? Because white matter has much wider myelin areas, correct?
Yeah, and so each poses its own challenges. It's actually easier to image gray matter because it's not so dense. But that color picture we showed at the beginning was white matter. we go to thinner sections and do blurs and tricks. So this is, if I just do a focus stack, you can see all the planes being focused really nicely. And you see places where axons are changing direction and going, branching to different directions. When I first heard about the rectilinear arrangement of axon tracks in the brain, I thought that doesn't make sense. Why would they do that? Why won't they just go like straight there, for curved or something? Like I said, I'm learning neuroscience. So I find that fascinating.
Have you ever found answers to that question?
Answering a question how is not so difficult. Why is a more difficult question to answer. No, the neuroscientists don't have an answer as to why it does that. It doesn't seem necessarily more efficient, but I think it has to do with something about developmental science and what's happening. Because if you study like zebrafish, you learn certain things happen and you work your way up in evolution, and things start to make sense when you study it from a developmental science point of view. But I'm not an expert on that at all. So quantitative Birefringence microscopy, what we do is we rotate a linear polarization, but we image through a circular polarizer. And so this is what happens when you're rotating the linear polarizer. The lines get dark and lighter in the background is gray, because there's always sunlight getting through a circular polarizer. But if you then plot the intensity as a function of the angle of the linear polarizer, you get quantitative information. The amplitude of these oscillations is linearly proportional to the density of the myelin, or how much is there over the path length of the light. And the phase of it has to do with the orientation of the optic axis. So you take this information and you do it for every single pixel in the image. And this is just the math relating to that. And you get a color image that tells you the orientation of the optic axis. This is probably a 1 by 2 centimeters. This is from dorsolateral prefrontal cortex. Okay, this is a section, there's an even larger section on the right. And what we're seeing here is a large bands of white matter tracks. So this is white matter, this is gray matter here on the edges. The integration time isn't turned up to look at right now. So we're looking at the orientation of the optic axis. And here you can more clearly see the gray matter, white matter on the right there.
Can I ask you one quick question? I think this would be a nice time to get into the topic of label-free imaging, because in brain we know that it doesn't come color-coded like this. So your scientists do some fluorescence techniques, some like GFPs or
exactly
tomato or whatever, right? But here you didn't do anything like that. So I think this gets into the label-free imaging. Do you want to talk about that a little bit? How you assign these colors?
Yes. So that's the beauty of Birefringence microscopy. It is label-free and it's wide field, which allows us to explore large volumes of tissue and get a bigger picture of where things are. And yet we do it at super high resolution because it's tiled. And so the color here is basically just false color that represents the optic axis orientation. Just telling you where the axons are going.
Yeah.
The orientation of the optic axis. And so you see the color change as the big bundles of white matter axons change direction. This is what's being recorded by the camera. And then you get the retardance map, and the retardance tells you how much myelin there is. That's the amplitude of that oscillation. And then you get the orientation map, and here's a key to the direction of the optic axis to pick out the direction of the axons. And this is in white matter, so it's really quite dense. You have to go to very thin sections. The sections are 20 microns, 10, 20 microns, but it's a pretty high resolution. And we can zoom in further to look at it. So I'm just going to go through a sampling of some of the images that my students have extracted for some of these studies. So this is in the boundary that's right at the edge of the white matter and gray matter, so it's actually fairly dense. but it's not as dense as pure white matter. And here's where you see these crossing. This is where this image is taken from, right?
Yeah.
You can see that some of the fiber tracks run parallel to the sulcus surface, and some are running normal. These are the diving axons, and I just think this is art. Prior student Nathan Blanke was able to generate some of these images, and this is taken in the white matter, but in an area where there's a bit of loss of retardance, probably having to do with the sulcus from below. You don't see the three-dimensionality to it. Different locations. Here's this location. This is lower resolution because I wanted to show how the axons work their way around the blood vessel.
Yeah.
You can see some tracks going diagonally one way, with the other way, but it's kind of cool how they work their way around the blood vessel.
So are you able to quantify in any way the myelin orientations, or is it just quantitative, qualitative, like looking at it and just learning from the images?
Let me get to that.
Yes.
Another brilliant question. Okay, so again, more examples of different locations. So here's what happens when an axon is transverse. Of course, all the colors appear because the optic axis is going around like that. And sometimes axons are coming at you. And so this is just adjusting the focal plane here. Okay, so here's what happens in disease. So this is normal healthy myelin. You see the optic axis is relatively consistent, running through the length of this thing. Here's a couple of transverse axons. You can see both in retardance and in quantitative. And that's what happens when myelin starts to degrade as a result of disease. I'm going to show you a lot more examples. So you can do a quantitative measure, for example, of the color variance. You can do statistical study of the variance of the orientation, as depicted by color, over a certain area. And then from that, you can teach an AI system to find flaws in the myelin. Okay. And that's one of the things you have to do, because we have so many slides and so much data to decipher, and we want to know something quantitative. Are myelin defects more prevalent in this stage of Alzheimer's and that stage of Alzheimer's, and in this part of the brain or that part of the brain? So we actually have to count them, and that would take humans years. So my students have become really good at training AI, mostly deep learning. Okay, so here's some examples of actual myelin defects and different types of defects. So this is what's left over when the myelin is broken down, and the lipid that's broken off just forms, lipids like to form vesicles. They just naturally form vesicles when they're surrounded by salty water. And you can see it's kind of a thinner thing, vesicle. There's no oil. But these are delaminations that are going on here. Swellings and delaminations. So it's a nice example there. And then there's the orientation map up there in the upper right-hand corner. And if you were to ask a neural network to look for, let's say, diversity orientation as a way of identifying flaws, that's one of the ways it can do it. This is just a clean, this is a CTE patient, and looking at a lot of gross breakdown of the myelin. This is an Alzheimer's patient. And statistically, if we count the prevalence or incidence of these defects in let's say, this is in the sulcus we're trying to stick with one part of the brain, so we're comparing the same region with every single patient and the same boundary between gray and white matter. And we see that the numbers of flaws between AD and CT and normal controls is statistically significant.
Yeah, it's interesting to see how, although there is degeneration in the myelin, it's vastly different from for Alzheimer's patients versus somebody who has suffered a traumatic head injury.
Yeah, there are differences in the type of my students are becoming really good at identifying different types of myelin flaws. So, for example, stroke, in the area around the stroke, even in part was not killed by the stroke itself, but the axons in that area generate a lot of vesicles, because the myelin just simply breaks off and forms vesicles, and then the body removes those. So that's why people partially recover from stroke. But if our colleagues at the medical center are developing treatments to amplify the repair, not just the removal of vesicles, but the repair of myelin that's been damaged by stroke. This shows that one of the things we're working on is because you only get contrast where the birefringence is, it actually has a pseudo-sectioning possibility to it when it's not too dense. So again, it's not confocal. There's nothing confocal about it. It's not true sectioning. But as you saw when we went through the Z planes, different planes have different things come into focus, and then the contrast is maximized when it's in focus, because all the rays go through that spot there. Yeah. And then you get the largest contrast because it's purely. So in fluorescence, that wouldn't work, because even if these objects are fluorescent in different planes, what happens is fluorescence, the background just adds monotonically. I have fluorescence from this plane and this plane, this plane, it's also going to add to the background, right? But in the case of birefringence, it's a question of the phase of the polarization angle. And that phase might go forward, might go backward if things are not on the orient. So it's not adding monotonically, it's just moving around back and forth, and that back and forth is sort of random. So it does spread, but it's not an additive thing. So it's not a linear additive thing. And so you actually get some sectioning properties to the image without doing anything. It just comes for free. And this is back to that pretty picture of white matter and really following tracks. So the people who are interested in the connectomics are also interested in this. Okay? So there are various people who have been coming to use our microscopes in our lab lately. There's a group that's been studying brain neuronal spheroids, organoids. Mm-hmm. And one of the markers of it actually becoming a viable organoid from brain tissue is it starts forming myelin. Well, like oligodendrocytes start myelinating processes coming from the neurons, even if it's only a little millimeter shear. But they have no way of imaging that. Now they do. So it's kind of cool.
Have you ever imaged with birefringence on living tissue?
Yes and no. Only the mouse sciatic nerve that we kept alive so we could image the propagation. But it's on our bucket list. We want to try to do this in reflectance mode rather than transmittance mode, which is not easy to do in reflectance mode, so we could image things going on, let's say, in the surface, in the cortex of a mouse model. So in addition to myelin, we have found that we can see changes in the structure of let's say, cerebral arteries. So these are different stages of Alzheimer's disease in cerebral arteries, which, because collagen and elastin and smooth muscle cells are birefringent, so we get images of them too. And again, it's not labeled, and it's wide field imaging. So just before he graduated, Nate made these images. But now we're collaborating with Catherine Zhang from the ME department and some collaborators in the medical school studying cerebral arteries. And one of my PhD students is working on that. And this is what, if you really zoom in, so here's something really cool. This is from a healthy cerebral artery of a healthy patient or a youngish patient. Okay, so one of the things that happens as you get older is your arteries get stiff, right? Atherosclerosis, your arteries get stiff. And because they get stiff, they can't expand. They're less compliant. They can't expand so easily on the positive pressure wave from the heartbeat. And so that restricts basically perfusion and blood flow to the brain, among other places, right? But a healthy, compliant thing has to be able to expand. So in order to be able to expand, it has to be able to stretch. And so here you see the connective tissue alternating color like that. It's sort of an accordion, because the collagen elastin layers, and my students know the names of these layers, something lamina, whatever, have this accordion structure that allows them to expand. In an older patient where this becomes stiffer, you see less of the accordion structure and more. Anyway, I don't do any of this. These are the brilliant students that do all the work.
Yeah, that is fascinating, truly.
Well, thank you very much. Now, viewers and us, we all have the idea how you got to the bigger picture that we have been seeing since the beginning. I know you briefly touched on it, but would you mind expanding more on, as a member of BU's Neurophotonics Center faculty, how does your work in optical diagnostics and biofringence everything intersect with the advancement that Neurophotonics Centers has been pushing over the years at BU?
Well, it's a question of sharing of resources and collaborating with people. So in the Neurophotonics Center, they have this polarization sensitive OCT, the serial sectioning thing, and we've done some really nice studies collaborating with the BOSS group in doing serial sectioning, so you get really large volumes of tissue, but at lower resolution, and then taking some of those sections and cutting thinner slices and imaging the same exact tissue with Birefringence microscopy. So in one case, it's like 5 micron resolution, in the other case is like 300 nanometer resolution, and getting the picture of when it's seen in PSOCT, what is the underlying structural changes that are causing those disease-related changes in what PSOCT measures. So again, polarization-sensitive OCT also measures retardance and can also measure the angle. And I didn't show any of that data, but we have this beautiful overlays. I would have to tell you which one was PSOCT and which was Birefringence microscopy, except that in the Birefringence microscopy one, you can zoom in and see individual axons, and that's the beauty. So we're learning to understand, and our students have just submitted 2 papers on that. We're learning to understand what are the underlying changes that result in the changed properties measured by in large volumes, and thereby how does that relate to disease state? And is it really have anything to do with amyloid beta causing the disease? We think not. Okay. And that's kind of being debunked lately, right? The amyloid beta and p-tau hypotheses. Not that they're unrelated, but it's not it's a question of what's causing what. So right now, you'll find just in the last 7 or 8 years a large number of publications addressing things like the sequence of loss of perfusion leading to oxidative stress on neurons, leading to breakdown of things that lead to that axonal degradation, which leads to myelin degradation, which we can see, eventually leading to other things that cause the body to deposit proteins there. Again, this is out of my field, but we have tools that can help people better understand, better know what's actually happening. And we're hoping to get some of that funded as well.
And piggybacking off of that, since we're nearing the end of the interview, we're going to transition into a quick Q&A section. But before that, one more question regarding future directions. So how do you think that the work that you have done throughout your career will develop in the future? And where do you see the field of biomedical optics going in general?
That's really a question for younger scientists. I like to put the seeds out there. Like I said, there are now there's 4 other groups that are sending their students to be trained on our birefringence microscopes and learning how to put circular polarizers in their conventional microscopes that there, but the quantitative measures coming, they book time in our scopes in our lab. So there's lots of directions that this can go. And so, for example, one of the things that will be going on long after I'm gone is developing treatments. So our collaborators at the medical center have, they're part of a partnership that's developed to have this monkey colony that they've had for 30 years. And so they developed very succinctly, accurately quantified stroke model and models for age-related dementia, and have been developing treatments. One of the treatments they've been published on is extracellular vesicles. Don't ask me how extracellular vesicles treat stroke or dementia, but they've shown that in the animal studies, animals that were treated either recovered faster from stroke or showed less signs of age-related dementia than animals that were not treated. So, keep telling them to make some pills for me really quickly. I'm getting old. But these are things that are going to go forward because this is very early stages, and I'm hoping that this kind of wide field, unlabeled imaging modality will become a mainstay of studies in neuroscience, rather than becoming, the new thing that people are trying to figure out how it works. Understand it, it'll become a mainstay. Will depend less on fluorescent tags and confocal microscopy. Will depend less on things. There's a kind of microscopy called SCORE, which can also image myelin, but unlabeled, but is basically a confocal technique, and so it's very slow. And yet, because it was unlabeled, lots of people started doing it as soon as it became available. Well, we think this is going to be even easier and definitely less expensive. The cost of instrumentation here is really quite a bit lower than it is for anything confocal. And if you don't have to deal with all of the procedures of tagging and labeling, it does become easier. So we've convinced a few of the neuroscientists in the medical center work with us, and now they're collaborating and also sending their students to develop new projects. So there are a myriad of directions this can go. I have a variety of other really pretty pictures that are taken with my Birefringence microscopy that have nothing to do that I'm pursuing, but it's just really cool. Anytime somebody has something, they just stick, can we stick this in your microscope and see what? So we occasionally get names on the paper here and there, just because they use our microscope. Questions?
Yes. So for the fun Q&A question part, we are curious to know what are some of the habits and practice that help you stay creative and motivated in your research?
Oh, I could go on forever. So I am very, very fortunate. I get paid a lot to do what I love to do. Okay? I'll never get rich as a university professor, but I'll never be poor. And I'll have a comfortable life. But I love my work. I love coming in to work, and it's mainly because of the interaction with the students. I love teaching. And for me, the most fun time of the week is my meetings with my PhD students, and I hear about what have they done in the last week and what are they planning to do, and then I throw a little bit of seeds in there or learn from them when we get to the point where they now understand what they're doing well enough that they know it better than I do. That's when it becomes really exciting. And the cross-pollination that goes on when you have a group of students where you talk about, well, this is the problem I've been having, it's not working, and somebody says, oh, you need to tune the thing that way, and the other students already know how to solve the problem. And so It's what gets me up in the morning. It's what brings me back here.
Now, let's just say that you have students that you're mentoring, that you love mentoring students. Are there any traits that you see that makes a successful PhD student who enjoys doing research, who enjoys getting a degree that you've observed?
You have to be curious. You have to be unafraid to make mistakes. It's sort of like the Silicon Valley kind of bear. Break something and learn from it. But so as long as it's not putting us out of business, it's okay to try things and end up, yes, we'll buy another filter if you break it. And I think the excitement about the underlying science sort of becomes contagious. And so I think in my nature, I'm more of an applied physicist, otherwise known as an engineer. An engineer who likes to understand the basic science. And so my nature is I'm always going to take a physics approach to the research we do, but it's got to have some application. It has to affect the human condition ultimately. And that kind of multidisciplinary science is so complex that no single person can have a handle on all of it. So you have to collaborate. You have to work in teams. You have to work interacting and stimulating and fertilizing each other with your world's most massive parallel computers.
Absolutely. And that's what Neurophotonics Center stands for. So that's wonderful.
What a wonderful environment for these kinds of collaborations.
I mean, we have also been enjoying, most of us are co-advised, so we work closely with two different labs, and we're seeing both the computational sides and the experimental sides, and just being part of the Neurophotonics Center and getting training in different types of microscopy and different types of data analysis methods has, at least for me, it has been tremendously helpful during my degree.
Absolutely. I'm also co-advised. Our producer is also co-advised, and a lot of the people in my cohort are co-advised. So it's wonderful. So for our last question, so when we asked you about the future of biomedical optics, you pointed at early researchers and scientists, and you have a tremendous amount of experience mentoring the students and all. And many of our listeners and viewers, I believe, are also early career research scientists, either doing their PhD in first, second years or thinking about PhD. So what advice would you give to aspiring PhD students or early career researchers who want to contribute to neuroscience using engineering and computational tools like you do?
At which stage in the grade?
Either first, second year PhD students or students who are thinking about PhD.
It's really important what environment you join to pursue a career like that. I like the model of PhD admissions that we have here at BU, where you do rotations in your first year and you're not locked into a specific lab the day you arrive. And so that gives both the student and the PIs a chance to get to know each other and decide if this is a good fit to the lab. You want to feel comfortable in your lab environment, and that in and of itself will help you be happier about the research you're doing. And also, again, since excitement about science is kind of a contagious thing, it's a contagion, and it's if other people around you are excited about something, you get excited too. But you go to a movie theater, if people are laughing, you may not even know what they're laughing about, because you didn't get it. You laugh too. Well, it's sort of like that with science. If you're excited about the science and you share that excitement, then others will be excited. So I asked a student, said, look, first of all, and this is what I advise my seniors. Do you want to go to grad school or do you want to go to a job? Okay? Right. So, do you like working 70 hours a week? Do you like working nights and weekends. Not because you're forced to, but because you're pushing yourself to do it. Do you like what you're doing to involve a sense of discovery? Are you willing to put up with long, frustrating periods of time when nothing works and you don't discover anything? If so, invest another 5 or 6 years of your life and go to grad school. If not, if you're an engineer because you want a good paying job and you want to have a life outside of work, then an industry job might be a good thing to do. And it's not a judgment, it's just different style. So here's an example I love to relate. A few years ago, I had 2 students who were finishing their PhDs within a month or so of each other. Okay, it's like 5 or 6 years ago. One comes in, I bring them in to sit down with them, a few months before, say, okay, what do you want to be when you grow up? I want to help you get there, okay? So one comes in and says, I want your job. You get to work on any science you like. You can change fields from this thing to neuroscience, and even though you don't have experience, you can do it. You can work on whatever you want, and besides which, the students do all the work for you. I want your job. Okay, you want an academic track. So I'll help you get a really good postdoc. The lab that'd be a great stepping stone, and from there you start applying to faculty positions after that. And then a week later, the other student comes in and says, well, what do you want to be when you grow up? I don't want your job. You never stop working. You send me emails that were sent at 2 o'clock in the morning and on Sunday night, and what? I want to have a life. So, okay, then I'm going to help you get an industry job where you work on projects with teams of engineers, and when you go home, it's your time. So both have merit, and both are needed, and both are important, but it's different personalities are different personalities. It takes different types of people to build a society, and all are either meritorious and worthy, and we need both. We need people in industry to develop our next iPhone or our next drug, but we need people doing the basic research to enable them to make the chip that does that, or to enable them to know how to manipulate the biomolecule to become a drug.
Yeah. Yeah, there's a lot of nuance, and as you said, it takes different personalities, and it's grad school is actually a good time for a person to learn more about themselves and to see what actually fits for them and what they would like to do. Okay, so on that note, Professor Bigio, thank you so much for coming on to our little podcast. It was lovely having you here.
Thank you.
Thank you for listening. If you like this episode and want more like it, please take a moment to share it with anyone you think would enjoy it too. Next time, we will be joined by Dr. Stefan Mihalas from the Allen Institute as we discuss his journey of studying the brain and thinking about how the noisiness of neurons is not a bug, but a feature of consciousness. This episode was made possible by the support of Boston University's Neurophotonics Center and listeners like you. Thank you.